But I'll introduce you in Portuguese. Please believe me, I'm not saying anything bad.
No, that's fine. It's just that, should we put the slides up before you make the introduction?
Matheus, põe os slides antes de fazer a introdução ou não? Pode,
depois,
né?
Depois acho que tá ótimo.
Okay. John?
Yes.
Actually not. Let me. I'm gonna say a few words and then you join. Okay.
Yes, but we haven't got the slides on the screen.
No. Yes. You have the time. When the time comes, you'll be able to share the screen. There's a green button.
Yes, I know.
Yeah.
That's
okay.
fine. Yep, that's fine. Good. That's fine. No problem.
Okay.
Thank you, Matthias.
So, yeah, thanks, Professor. So we are live, so we can start. Thank you so much.
Yeah, we are live?
Yeah.
Boa tarde, pessoal. Bem-vindos de volta ao nosso segundo dia do evento do PIBIC. Tem sido absolutamente um sucesso o engajamento dos alunos na visita dos painéis. Já tivemos mais de 23 mil visualizações dos posters, dos vídeos, enfim, do trabalho dos colegas. Tem sido absolutamente maravilhosa a resposta e a participação de todos vocês, de toda a comunidade. É uma surpresa muito agradável. Nesta tarde, nós teremos o prazer de ouvir o professor John Pickett. Ele é da Universidade de Cardiff. Como vocês sabem, talvez não saibam, mas a Universidade de Cardiff e a Unicamp estabeleceram uma parceria estratégica. Nós, na verdade, cada uma das duas universidades coloca um pouquinho de dinheiro e a gente envia alunos, doutorandos, pós-doutorandos, professores, de lá para cá e daqui para lá todos os anos, e tem sido absolutamente maravilhosa esta colaboração. O professor John, ele completou a sua graduação e o seu doutorado na Universidade de Surrey, na Inglaterra. Depois ele fez um pós-doc na Universidade de Manchester. Ele atua na área de química ecológica. Ele tem trabalhado intensamente em estudar as interações quimicamente mediadas entre os vários organismos, inclusive as pestes, né, as pragas, digamos assim, que atacam animais e plantas. E ele usa esse conhecimento para desenvolver o que se chama de ecologia química. Então, o professor John Pickett é recipiente de inúmeros prêmios. Ele ganhou já o prêmio Rank da Sociedade de Nutrição e Cultivares. Ele é membro da Royal Society. Ele é membro da Academia Alemã de Ciências. Enfim, ele foi nomeado Cavalheiro do Império Britânico na área de Química Biológica. Enfim, ele é recipiente do prestigiadérrimo Wolf Prize na Agricultura em 2008. Enfim, as contribuições do professor John Pickett são absolutamente gigantescas nessa área e será um prazer ouvir o professor John nesta tarde. Então, Professor John Pickett, it's a great pleasure to have you here with us this afternoon. And the floor is entirely yours. And afterwards, I'm sure people will send us questions. We've been streaming live on YouTube. Thank you very much. Please go ahead.
Thank you, Munir. Let me share the screen. Can you see that?
Yes, John, yes.
Right, I'm going to maximize the screen. There we are. Is it still visible?
Yes, it's perfectly visible.
That's fine. So thank you very much for that wonderful introduction, which I regret I didn't understand all of, but it sounded very nice. It's also sounded very accurate. So I'm Professor John Pickett, and there is my title. And there is a picture of the front of the university in rather better times without people wearing masks. And on the right is the picture of the chemistry building next to the administrative building. The subject of my talk today is the pheromones, the chemicals that are used for communication between different organisms of the same species. It's a term reserved for communication between members of the same species, but unlike this definition here, it doesn't just relate to animals, it relates to all kinds of organisms, including plants. We have a very clever way of listening in to what the insect is hearing when it hears a pheromone in the air. And of course, it doesn't hear it, it smells it. And the smelling is done with the antennae. And there on this beautiful sphingid moth, you can see the antenna with electrodes in it so that we can record from the antenna and record what it responds to when we separate complicated mixtures of the pheromones mixed up with everything else that smells in the air by gas chromatography. We can either couple the gas chromatography to the electroantennograph, the EAG, or a single cell or single neuron recording. It's been a very valuable tool, and over the years we've identified many different pheromones from different insects and indeed other animals. Some of these are moths, most of them are moths, and at the top there's a beetle. If you look at the very small moth, the Phyloneuricta moth in the middle, it's almost difficult to see, certainly flying around at night. When we take the volatile chemicals from the air, when it's releasing the pheromone, we get a number of peaks, as you can see on the bottom trace here, of the flame ionization detector of the gas chromatograph. But at the top, we have a wonderful trace, which is a recording from the whole antenna, the electroantennograph. And that massive deflection in the standing potential that we record as the electroantennograph shows us exactly where on the gas chromatography trace our pheromone is eluting. And that means we can do the same experiment on the gas chromatograph coupled to the mass spectrometer. And that gives us the spectrum, which allows us to tell the compound. And you can see it there, fully identified as Z10 tetradecynyl acetate. That insect is a pest and does a lot of damage to trees. They also damage woodland trees and fruit trees. Here's another pest of not quite a tree. It's the banana plant. And the banana weevil, Cosmopolites sordidus, is a major world pest and a very important pest in Brazil of your Brazilian banana fruit products. You can see a farmer there with a machete, or panga, as it's called in one of the African languages, and that's about the best thing you can use against the banana weevil if you can't afford insecticides, which is mostly the case for poor farmers in Africa that grow bananas. Here's our trace again then with the gas chromatography flame ionization trace at the bottom, and at the top we've got the gas chromatography coupled to the electrophysiological response of the insect's antenna, that beetle's antenna. And you can see a big peak there, but when you follow it down to the chemistry trace, it's a really very small peak. And in fact, we've had to mark where the peak is by the time, 11.93 minutes. That's how long it took to come out of the gas chromatograph. But when we put that onto the mass spectrometer, the molecule breaks up by a process called the retro-Diels-Alder reaction. And that gives us a definitive identification of the compound. And I've drawn the structure out there for you. But this structure is very sophisticated. It contains 2 carbons asymmetrically substituted. That means that they can exist as a right-handed or a left-handed atom. So at carbon 2 and carbon 5, we have this very clever stereochemistry, right-handedness or left-handedness, which gives rise to 2 to the 2, 4 isomers of the compound. And it's very specific which the insect responds to. At the top trace, we've got the electrophysiological trace, and then below it, we've got all 4 of the isomers. But when we look at the natural product, which is just a shoulder on that peak that you can see there, we only get 1 response from the antenna. And that tells us exactly which stereochemistry we've got. And it's the 2R5S that is the pheromone of this animal. Now we turn to a major arable crop pest, the aphid. There are many species of aphids, and back in 1987, we published in Nature on St. Valentine's Day the first identification of an aphid sex pheromone. The media thought it was very amusing to have the sex pheromone of an insect on St. Valentine's Day. They, of course, didn't know much about insects because they've shown the aphids, which are insects, with 4 legs. They, of course, have 6. The pheromone is used by the males to find the female, and you can see males with the female now arriving there by being attracted to her with the sex pheromone, which is those 2 compounds there, again with some asymmetric carbons. This time, 3 asymmetric carbons for the compound on the left and 4 for the compound on the right. Very sophisticated chemistry, and the aphid knows exactly what it is. We found the compounds by now doing single cell or single neuron recordings. There we have a cartoon with the electrode placed close to the cell body of the olfactory neuron in the organs on the 3rd segment of the male's antenna, which we already knew were responsible for detecting the pheromone. There's the trace from the work in 1987, and 2 increases in spike potential, spike frequency, showing us where the pheromone compounds elute. So we did the mass spectrometry, and there's a picture of the aphid showing its legs in the air, and it's from its legs that it releases the pheromone. We capture those chemicals from the air on a porous polymer, run them through the gas chromatograph with the aphid joined at the single cell or neuron preparation. These are the many papers that came out of that first paper in Nature, until we come to some new work, which I'll talk about in a moment, on synthetic biology, but relating to what aphids can smell. But there are the pheromonal compounds of most of the pest aphids. And you can see they all have this clever and sophisticated stereochemistry by having right-handedness and left-handedness in the carbon atoms of some of the carbons in these molecules. There's the P aphid, Acyrthosyphon pisums pheromone, with the 2 separate cells firing, one to the lactol and one to the lactone. And in 2010, we and many scientists from around the world published the full genomic sequence for this aphid. And that's now allowing us to deduce how the aphid biosynthesizes these chemicals and how it responds to it. And we're soon to publish the work that Suzanne, you can see pictured there, did for her PhD at my old laboratory before I moved to Cardiff, there, Rothamsted Research. I'd now like to turn to some other chemistry that the aphid responds to, but this time it's from the plant. And when aphids feed on the plant damage causes the plant to make some chemicals which would frighten away other aphids and which can attract in insects that attack aphids, insects like ladybirds and parasitic wasps. If we now look at the gas chromatograph and the electrophysiology, we can see a chemical called S-germacrine giving a response. And that is one of these chemicals produced when the aphid attacks the plant. So the plant calls out to the aphid to go away, but it also calls out to parasitic wasps and ladybirds to come and attack the aphid. Now, we found out what that compound was using the electrophysiology. And then at Cardiff, with my colleague, Professor Alleman, we take the molecule that we know the aphid uses, and there it is on the right, S-germacrine D, and the R's are hydrogen atoms when it's the native compound. But we're trying to make analogues that might be more active and more stable. And so we take the precursor, the farnesyl diphosphate that you see on the left, and we let it be converted into the semiochemical, the chemical from the plant, by the germacrine D synthase enzyme in the presence of magnesium, which is a cofactor. And we argue that if the substrate, the farnesyl diphosphate, can be substituted at the R positions, then we will get new compounds. And if they're converted into the kind of germacrine that we have here, then that will give a response to the aphid. And that indeed works. And we've been able not just to make attractants, but also repellents. And we've been able to develop these against aphids in a push-pull technique, which we're now field trialing in Spain as soon as we can get out into the field in the new year with colleagues from Wales and colleagues from Spain into the lettuce crop. We also have a project working against whitefly. And there we are with the now modified substrate, the farnesyl diphosphate. Again, it's got different stereochemistry, but it makes epizingibberine, which is a very, very powerful repellent of the whitefly. And we're hoping we can do the same with the chemistry of that as we've been doing with the aphid chemistry. I'd like to go to Africa now, because we've been talking about using the pheromones and the other signal chemicals, the semiochemicals, made by synthesis. But now I'm going to talk about using the chemistry of wild plants and commercially useful plants to produce semiochemicals that will protect crops against pests. And we're talking about a process called the push-pull, or in American, stimulo-deterrent diversionary strategy, or in one of the African languages, pull-push, vutasukuma. And here we grow plants between the crop, maize perhaps, or sorghum, that both attract the natural enemies of the pest, but push away the moths themselves. We then attract the moths into a trap crop where they lay their eggs. And we feed the trap crop to cattle, and the cattle eat the larvae with the forage from the trap crop. One of those intercrop plants that does this magical job of attracting enemies and pushing the moths away is a grass called Melanis minutiflora. And that makes a smell that's just like the smell, to the insects anyway, that maize produces when it's attacked by insects. And it attracts the parasitic wasps like you see there, Cotesia sesami. And those two publications there explain how we found with our African colleagues, working in sub-Saharan Africa in poor situations where farmers can't afford pesticides, to try and use this chemistry, but delivered by the intercrop to control the pests. There's the intercrop, the molasses grass, and you can see it's making that compound that really is very good at attracting the wasps to attack the moths from a higher member of the isoprenoid class of natural products, nerolidol. It cuts off 4 carbon atoms. When the plant is attacked, it cuts off 4 carbon atoms, makes that compound at the bottom, the nonatriene, and that does the job for us. We've even found that when the moths lay their eggs, the plant detects the eggs. And when it detects the eggs, it starts to produce that chemical even before the insects have arrived as larvae to chew the plant. It attacks the wasps attack the eggs as egg parasitoids, and they attack the larvae as larval parasitoids. A very clever job, which we call smart plants. But it's not the main hybrids of maize. If we look at the electrophysiology again, there's one of my colleagues who does the electrophysiology, and there's one of the students, now a lecturer in his own right on the project, and there's the detector's response at the bottom with the red dots over the compound we've already seen, and another relative of it with 5 carbons more. 5 carbons more because it's a higher homologue in the isoprenoid pathway. And that really does such a great job that we got a lot of different funding agents to join us together with African farmers like these two here, and African scientists like Damaris Odeni there, and an American maize geneticist, Ed Buckley, you see there. And we started chasing the genetics in the not hybrid maize, but in the landrace maize and teosintase that the maize has been developed from, to find the genetics so we can breed it in even more powerfully. We find that the chemistry that switches on those genes is associated with the eggs, not the chemical I've shown there, which is part of the lipidal glue that holds the eggs on the plant. But with the knowledge of that chemistry, we can now chase the genes more readily. That's what the whole push-pull looks like, with another trap crop in between the maize, and that's a forage legume called Desmodium, and then the trap crop on the outside with the maize growing in between the intercrop, protected from the insects. But it's also protected from a parasitic weed called striga. And there we can see the control of the farmer using best practice in maize and in sorghum. But where we've got the push-pull system, then we control the pests, and so the farmer gets a much better crop. And that difference in crop yield, although it's very small compared with the kind of yields you can get on farm in Brazil, for example, then that makes a great deal of difference to the economy of those poor, resource-poor farmers who can't afford insecticides normally to control the pests. And we've been working on drought-tolerant push-pull, and there's some of the intercropping plants that push the pests away and attract in the beneficial insects and damage the pest weed so that it can't damage the crop. And these are drought-tolerant plants, which is allowing us to develop a drought-tolerant push-pull system. Back in 2014, we told the world in this publication that we were going to have over 1 million sub-Saharan African poor farmers in the push-pull innovation by 2020. Well, we've just about managed it, though it was very difficult with the epidemic around the world of COVID-19, but we were helped by our push-pull controlling the fall armyworm. Now, the fall armyworm is a major pest in Brazil and in North America. It's recently arrived in Africa. It's moved on to India and to China. It's doing untold damage, but the push-pull works really well against it. Here are some statistics from one of our publications on this, and the red shows the normal maize crop and the very large amount of damage which the farmers are getting, largely in industrial production, because it's the smallholder farmers that have the push-pull. And the blue signs are the very small numbers of pests, the fall armyworm that they have in their maize. So that's really worked very well. And of course, it's pushed up the number of farmers who are interested in it. The other way of delivering this kind of chemistry is by GM, genetic modification, including genome editing with CRISPR-Cas9. Well, we don't have that nice compound we saw a little bit earlier that we were using in Africa with all the genetics sorted out yet. But we do have another chemical which repels aphids. And it repels aphids because it's produced by the aphid itself when it's attacked. You can see the aphid being attacked by a predator, and that little droplet there is a sticky defensive secretion. But evaporating from it is the alarm pheromone, which comprises another isoprenoid, a C5 compound with 3 of those groups. It's a sesquiterpene called E-beta-farnesene. We knew it would work against aphids because this wild potato from Bolivia, Solanum batholtii, has special hairs on its leaves which make that little droplet like the aphid does and release the pheromone. And that's back in the days when you could publish in Nature with just 2 authors. Now we publish in Nature with lots of authors. We wanted to capture the genetics from plants, but when plants make E-beta-farnesene, they also make some chemicals by the A roots here, like caryophyllene at the bottom of the picture. And unfortunately, when the aphids smell the caryophyllene on their antennae, they don't respond to the E-beta-farnesene. So we had to do some real genetic engineering, because we also knew that the predators and parasites of the aphid, they could also smell caryophyllene when it was produced with the alarm pheromone. And so they wouldn't go looking for aphids and eating aphids. But engineering the genes allowed us to demonstrate in the model plant Arabidopsis thaliana that we could produce the E-beta-farnesene more or less without the caryophyllene, which in fact is produced naturally by Arabidopsis thaliana. Then we moved to wheat, major crop. The first construction we had to make the E-beta-farnesene, the E-beta-farnesene synthase, didn't work. So we added another one then, the farnesyl diphosphate substrate. So we made more of that in the wheat. But as you can see from the crosses there, that didn't work. But when we managed to target this enzymic apparatus to the plastid in the wheat plant, then we got a low level of E-beta-farnesene with just the E-beta-farnesene synthase, and a higher amount, about 10 times as much, with the farnesyl diphosphate synthase as well, all in the plastid. And this was really very good. In the lab, we could use our GM wheat to frighten the aphids, the cereal aphids. We could also increase foraging by the parasitic wasps, which lay their eggs in the aphids. And you can see the picture there, so that the GM wheat is now pulling those wasps into it. But that was all in the lab. We had to go into the field. We had to surround our crop with a big fence so that the genetic material couldn't be taken away by people or animals. And we separated the plots of GM and control wheat. The GM wheat had the low level or the high level of E-beta-farnesene releasing. And then we had barley as a barrier crop. And then we had more wheat as another crop. And we did the experiment in 3 seasons. But unfortunately, as you can see in that paper there, it didn't work. It didn't control the aphids. We produced the pheromone as we'd intended with the 2 different levels. The genes all worked, the wheat worked, it produced seed, it set seed very nicely and proved a useful crop producing plant, but it didn't control the aphids. And that's because we didn't imitate the way the aphid produces the pheromone. And so we're now trying to do that genetically with a colleague of mine at Aberystwyth University, also in Wales, Hugh Jones. And there's some genes there, the middle one, which is switched on by aphids very powerfully, which we identified with some Chinese colleagues. And we're now trying to use that to make the aphids frightened by the E-beta-farnesene being produced in a sudden burst. I'd like to now turn to some work that we've been doing with Brazilian colleagues. Clara Beatriz Hoffman Campo is the supervisor of Salvador Lima, is now got a nice job in industry. And she works on an organism called Phacopsera pachyriza, which is the causative agent of Asian soybean rust. And you can see the soybean crop in Brazil there afflicted by that fungus. And that fungus could take out not just the soybean crop in Brazil, but in the entire world. It's not transmitted by insects, but we were interested to see if the plant, like it can detect attack by insects, could detect an attack by Phacopsera pachyrizae. And indeed it can. Here's some pictures now from Salvador Lima's PhD, in which he actually shows that this compound related to the aphid alarm pheromone, but not the same, it's a different isomer, but it's produced by the plant normally. But when the plant has got an infection of this fungus, it stops to produce it. And Salvador found this, and Clara Beatriz Hoffman and her colleagues in Brazil, we've been looking at the genetics, and we've just this very moment published a paper on that. Now we're trying to build this into plants so that the plants can actually switch on a color, which you can see this oilseed rape flower doing, so that it picks up the color when it's infected by the soybean rust. And of course, either you can see it from a distance with binoculars, or you can send a drone in there to see it. And then you know when to spray the crop with a fungicide, because it develops very, very fast. And this loss of this E-alpha-farnesene takes place much faster than you can use molecular diagnostics or tell that the plant has got this infection. I'm going back to Africa now to look at an animal problem there, which allows me to come back to Brazil, to some other animal problems that we've been looking at here with colleagues. This is a cow with a disease called Nagana. It's caused by animal trypanosomiasis. The trypanosomes are living in this animal, depleting its production of meat and, of course, milk. But this animal is in the same family as the cows, the Bovidae, but it's a waterbuck. And the tsetse flies, the flies that carry the trypanosomes to the cows, never, ever bite the waterbuck. And so we found out with our African colleagues what the smell of waterbuck is. And there is the chemistry. And the chemists among you can work out what those compounds are. But we won't go into that, it's published. But mixing those four chemicals allows us in field trials in this region in Africa, where each red dot is a small group of cows owned by the smallholder farmers that live in this region of Kenya. And they have used on their cows a slow release of those four chemicals. It's a rather expensive and cumbersome device, but it's just an experimental one. But that, combined with a flag, blue and black, which attracts the insects, and it's got an insecticide on it, which kills them. So we can push with the smell of the waterbuck, the flies from the cow, and drive them towards the flag, which they see and like to go to, where they get killed with the insecticide. And that all has conspired to reduce not just the number of flies attacking the cows, but the flies that are transmitting the trypanosomes to the cows. And the thick line there is the push-pull, and does really very well. And we're now making much cheaper devices so that very poor farmers can avail themselves of this. Now, coming back to Brazil, I have a wonderful colleague at the University Federal in Goiás, and that is Dr. Lydia Miranda Ferreira Borges. And she came to me at a meeting one day, a long time ago now, and said that she found that some dogs didn't have ticks. Could it be that the dogs smelt differently? And I said, yes, I'm sure it would be. And so with Lydia and her colleagues at the university, we started to look at the beagle dogs, which were the ones that smelt so that the ticks didn't stay on them. And that meant that eventually we could take two of the compounds responsible, two simple compounds, just 2-hexanone, the ketone, and the aromatic aldehyde, benzaldehyde, and in a slow release collar, put them onto the English Cocker Spaniel, which gets very, very large numbers of ticks, and immediately reduce the tick numbers down like the beagle. Working also with Ligia, we've looked at the difference in smell between donkeys and horses. Horses, very valuable animals, of course. Donkeys, useful animals, but not afflicted by ticks and other arthropod pests. And they use this simple compound, the 2-octenal, the C8 unsaturated aldehyde. And when you put that onto the horse, then that frightens away the ticks. And Ligia is working with vets now to try and exploit that in Brazil. Now, what about ourselves? Finally, in the last few minutes of my talk, I'd like to turn to the mosquitoes that attack us. This is a member of the Culex genus mosquitoes, Culex quinquefasciatus. You'll know this mosquito because it's a vector of the parasites that cause West Nile virus, but also Zika virus. Well, I was the first person to identify a mosquito pheromone. Brian Lawrence, my colleague, is a biologist, so he couldn't identify the chemistry, but he helped me identify the chemistry by giving me samples that contained the pheromone. And the pheromone is produced by the eggs of the mosquito when it lays them on water. And that's the compound there with 2 asymmetric carbons. 2 to the power 4 gives 4 isomers. And it's only that isomer that works. Well, we moved to Brazil to try and develop a way of producing that pheromone rather cheaply. In America, it's sold for people to keep the mosquitoes out of their patio, but it doesn't find much use in poorer regions where you need to spend a lot of money on the pheromone if you're going to kill enough of these mosquitoes to stop Zika virus transmission. And so with a colleague from the University of Maceió, Professor Antonio Eusebio Santana, a very good friend of mine. We like to have a beer together when we can. And I'm hoping that when the COVID-19 is finished, I will be able to go to Maceió and enjoy some celebratory drinks with him. And with various people, we've been working on the sandfly pheromones. They are horrible insects which cause a very nasty bite, but they transmit leishmania, an appalling parasite that causes visceral, cuticular, and mucosal leishmaniasis. And that's a map of Brazil. I hope you'll excuse me, there's no other countries there. And 3-4 regions we've been working on the pheromone from. And from Lapina region and from the Jacobina region, we've got those 2 chemicals. The Santarém is not quite right and the Sobrel is not there. But those chemicals are pheromones of the sandfly, Lutzamia longipalpis, that transmit the leishmaniasis pathogens, the leishmania. We can synthesize the Jacobina type of pheromone, and we use a Diels-Alder reaction. We first of all started a late stage Diels-Alder reaction, but then we realized we could get a better yield if we did it first of all. So if you go to the bottom of that slide, the chemists among you can see something set up in the middle of the bottom line for a Diels-Alder reaction. And then that gives rise eventually to the pheromone with lots of stereochemical centers. You can see 3 asymmetric carbons there, giving us 2 to the power 3, which is 8 isomers, but only 1 for the sandfly. Now, this is where we worked with Professor Eusebio Santana, because we found that a plant growing quite easily as a garden plant normally produces the chemical there, germachrone, and we could work that through to give us the pheromone. And Eusebio, with his colleagues, has managed to start to produce that on a commercial scale to produce the pheromone for trials. The sandflies use the pheromone in a kind of lecking behavior, a swarming behavior. And we think, although it's produced by the male and affects the behavior of the female, whereas normally pheromones, sex pheromones, are produced by females to attract the male. Here we have a pheromone produced by the males, which influences the behavior of the female. And we think we can use the lecking behavior, the swarming behavior, to get better control of these insects, initially with insecticides, but eventually with more mechanical and more sustainable means. And together with another colleague of mine at Nottingham University, we're working with Eusebio, who we've already seen, to try and use the genetics of the Lutsamia longipalpis to identify the genes by which it makes the pheromone, so we can make the pheromone much more cheaply in a fermenter. Back to the mosquitoes then. Here's a young man that did his PhD with me quite a long time ago. He's now Professor James Logan at the London School of Hygiene and Tropical Medicine. And you can see there from the publications that we've been working on the heritability of attractiveness to mosquitoes. And that's very interesting because some people are just not attractive. And they produce some interesting chemistry, which is repellent to the mosquitoes. We wonder if we can perhaps use it to repel the mosquitoes. We haven't done so yet, but we're looking at the heritability, hoping that we can develop a kind of cream for people who are very susceptible to being attractive to mosquitoes, put it on their skin so they make it into the repellent, a kind of slow way of releasing it. We've also been working on what happens, particularly to children, when they get infected with the malarial parasite. So the malarial parasite is transmitted in sub-Saharan Africa, where these children live, by the mosquito Anopheles gambiae. And here's the really nasty part. Although we, as soon as we detect malaria in the children, we cure the malaria so they don't develop the disease symptoms. But the nasty thing is that the plasmodium, the pathogen that causes the malaria, makes the children more attractive to the mosquito. It causes the children to make chemistry, which you can see on the bottom of the slide, that attracts in the mosquito so that it can take a blood meal off the children and transmit them the malaria parasite to them. Now, isn't that really nasty? But now we know what the chemistry is, and even some of the genetics on how it's making the compounds. We can start to use this against the malaria mosquito, and perhaps also against the plasmodium that causes the disease. Now, all this is about changing the smell of human beings. And if the human being catches malaria and changes the smell, like the children have done, then maybe, and we think it's almost certain, that people with SARS-CoV-2, the virus that causes COVID-19, we're pretty certain that that makes people smell differently. And so we're using the same kind of approaches to identify the chemistry of the smell of COVID-19. And we've already got dogs that can do that, which we're training to be a very first layer of detecting the disease in a non-invasive method. So let's see how that develops. That's my work for the future. Thank you very much indeed.
Professor John, thank you very much. It's a splendid lecture. Thank you very much. We have a series of questions. People are reacting very positively, very well to your talk. I'm sure we have a few minutes for answering questions. I'll start with questions. One question from Mateus Fontanari. He's a student of production engineering, and he asks whether repellents produced from pheromone analogs, whether they have any environmental impact.
Well, it's a very good question. Personally, I think I'd prefer to use the natural pheromone because that's there out in the environment. Often we're in touch with it ourselves. They are very, very benign chemicals. And I think it would be safer in the long run to use the natural products. But, you know, we live in a world where the pharmaceutical and agrochemical companies like to have patent protection, and they like to get more active chemistry than is in nature. Personally, it's my view that it's very difficult to get chemicals as attractive or as powerful repellents as the natural ones. So there's a battle on at the moment. I'm caught in this battle myself, really, because as a chemist, I like to do both. But really, I think my heart is in using the natural products delivered by fermenter technologies, GM plants, or by synthesis, but certainly as the natural products. So yes, it could be that they are not as benign as the natural products, so we should certainly be very careful. Next question.
Hi, Professor.
Hello, Daniel.
I work with Professor Munir.
I'm so sorry. I'm so sorry. I was muted.
Yeah, he's back.
I'm sorry. I'm back. I'm sorry, John. Okay, let me rephrase the question, okay? I'm sorry. Marcos Beccaro, he's a student, a master's student in economic sciences, and he asks if it would be possible that with time, the pests with less attraction to those pheromones, they could be selected by nature.
That's an absolutely wonderful question. I must say, Chair Munir, you've got some very, very clever questioners here. Yes, well, the day after tomorrow, I shall give a lecture, an entire lecture on that subject. And of course, I have an answer. The answer is that if an insect becomes resistant to our use of a pheromone or a repellent, then it probably has to use just a different chemical, because it must use the repellency to avoid danger, and it must use the pheromone, where it's an attractive pheromone, to find a mate, for example. It still has to do that. We've already seen evolutionary processes at work with pheromones, and there are a number of publications which show that insects gradually move away from the original pheromone. It allows speciation. It's a kind of process which involves different production as well as different detection mechanisms evolving simultaneously by natural selection or by forced selection, which is what we're talking about if we cause the insect or whatever it is to become resistant to the pheromone or the repellent. But unlike an insecticide where you have to go and make a new one, not just another insecticide like it, but a totally different insecticide to avoid the resistance mechanisms, here we just have to find out what the new pheromone component is or what the new repellent would be, and then we can replace it in a resistance defeating strategy. So by following this evolutionary process, we can perhaps beat the pest. But furthermore, and this is my other suggestion, which I'm talking about in 2 days' time, maybe we can use evolution to actually design something that we've already got ready to overcome resistance. So we use evolution, forced evolution, in our favor. But we try and do the genetics in microbes, which can go through many, many more generations than, of course, insects or other animals. If you send me your email, I'll send you the paper that I have recently published on this.
We'll definitely get Marco Beccaro in touch. Next question is from Sil Alves. She is a medical physics student, undergraduate student in medical physics. And she would like for you, Professor John, explain a little bit more the question about the pheromone problem that you talked about in the soy crop fungus. Was not clear
Yes, it's a very complicated story, because when plants are attacked by insects, they produce extra compounds. In fact, we think that the extra compounds that human beings produce, which repel mosquitoes, so some people are less attractive than others, we think those extra compounds actually relate to some kind of stress, even though the people who are repellent to mosquitoes are quite normal in their behavior and indeed in their smell to us. But with the soybean, Glycine max, it's different. When the disease starts to develop at a very, very early developmental stage, before you can see the lesions in the leaves, and before you can use molecular diagnostics, the plant starts to stop producing this E-alpha farnesene. It reduces its production. But nonetheless, we think that the process, the genetic process, could be used to trigger the production of anthocyanins, which are the purple colorants of plants that you saw in the flower of my slide with the drones. So I can understand why it was difficult to understand. I'm sure I didn't explain it well enough, but certainly it's complicated because it's the other way round from the insects, that when they attack the plant, produce the chemical. In this case, the plant reduces chemistry when it's attacked by the fungal pathogen.
Thank you. Another question now from Eduardo Teixeira. He is a student in agricultural engineering. He also has a question regarding the problem of the wheat. It was not clear to him how the way that the molecules produced would affect the result of the research. That was the question that, if you could...
I understand. So what we were doing, we were genetically engineering the wheat to produce an alarm pheromone that the aphid uses. Alarm means it's used when it's attacked or frightened. So when the aphid is attacked by a ladybird or a parasitic wasp, then it produces that sticky secretion you saw, and the pheromone, which warns the other aphids that they're being attacked. So we put it into the wheat plant, hoping that just like the Bolivian potato, with the special hairs on the leaf that frighten the aphid by producing a mimic of that pheromone, we wanted the wheat to do the same, to frighten the aphid away, to make the aphid think that it was being attacked, and that some aphids that had already been attacked were releasing the alarm pheromone. We certainly managed to produce a wheat that by genetic engineering could generate the alarm pheromone, the E-beta farnesene. But sadly, we didn't produce it as a quick puff, a burst of the pheromone, which is how the aphid makes it. And that's why we're doing the new genetics with genes that are rapidly expressed when aphids land on the plant, so that we can use that to cause the E-beta farnesene to come out of the plant as a burst, and we hope then frighten the aphid.
Thank you very much. I have a question myself regarding the mosquitoes. Mosquitoes, they attack me very much, but they don't go after my wife at all. She always claims that it's because I stink. So I think she's right after all, isn't she?
Well, your wife is not attracted to the mosquitoes. So it's actually she who produces the extra chemistry. I mean, I'm sure your wife is more attractive, or at least as attractive as you, Munir, but the fact is she's producing extra compounds which put the mosquito off. Now, there's nothing wrong with your wife, but the mosquitoes are very, very sensitive to how the host, the potential host, might behave. They really don't want to be standing on somebody's wrist if they are not going to be a good blood meal, or if they're going to swat them. And maybe this chemistry relates to some behavioral trait that was possibly not even in human beings as they exist now, but in an evolutionary stage earlier. But what we don't quite know. but certainly the chemistry that is produced almost certainly by your wife. And if we were allowed to take some volatile chemicals from your wife and look at these chemicals on the GC mass spectrometer, I'm sure we'd find one compound called geranyl acetone. And that's what helps your wife have this very, very nice trait. And quite a lot of people have it. Doesn't matter whether they're black, white, women, men, doesn't matter. Some individuals have this trait and most individuals don't. And we've been working on people with that trait. And at the moment, we have a large number of twins in Britain and in Africa. And we're looking at the fact that it's an hereditable trait. So if you trace it back to your wife's family, they will find where they've got the trait from. She's a very lucky lady in many ways, Munir.
Well, I guess the mosquito will just tell, let's go for him. He's the slow one.
That's possibly it.
Yes. Two last questions, perhaps. If Raphael, he doesn't mention what course he's on, but he asks if the mosquito can detect and avoid humans that carry on pathogens.
That's a very interesting question. Well, for some pathogens, they go preferentially to the humans. And that was the problem for the poor children that you saw in the picture. When the malarial pathogens started to develop in their bodies, they became more attractive so that the pathogen would be more readily transmitted. So although you might think the mosquito doesn't want to go on an unhealthy human being with a disease, well, it seems to look around for that, but it doesn't do it intentionally. It may even be disability to the mosquito to carry the pathogen, but it's compelled to go to the children with malaria because of their smell. It's using the smell that it finds its hosts with against the mosquito, as well as probably against us in the benefit of the pathogen. It's a very interesting question, but it can be reversed in some situations.
Thank you very much. I'll have one last question. Now I'll pass it on to Daniel Martins de Souza. He is a faculty member at the Institute of Biology, he works at the Vice Presidency for Research Office as well. Daniel, please.
Thank you. Professor Pickett, thank you for the nice talk. Very interesting, wonderful. So your research actually showed us how wonderful nature is. I'm a biologist, and I'm really amazed when I see this type of data, because we see that how nature evolved and is somehow controlled, and how the chemistry behind it happens. You've already said that there are some modifications that are done on plants, so we can, for example, have some of those vegetal pheromones, for instance. But could we think about genetic modifications that go across species, saying, for instance, having animals that can produce these pheromones, plant pheromones, or is this too far from the reality?
No, I think that if we identify the genetics in the human beings, although we wouldn't want to make a transgenic human being by any means, I think we might want to make a transgenic chicken. And I already know that if you feed chickens on some plants, they become repellent to the poultry mite, little red poultry mite. And that's a very big problem across the world. And it might be possible to take the chicken and allow it to make some of the plant compounds, which this carnivorous mite, this small arthropod, really uses to find the chickens. It comes out of its hiding place at night when the chickens roost and gets under their feathers, particularly under the wings, and starts to feed on their blood. So that might be a test for this, first of all. But I'm very careful to moderate what I say and do, depending on how the public view it, because eventually they have to eat the chicken or eat the eggs. And if they don't like the idea, then that's not really very good. But of course, as you know, we have to use technology as best we can to increase food production. And in increasing food production, we should do it sustainably. And that means we shouldn't use pesticides for which we get resistance and then have to spend nearly $1 billion making a new insecticide to replace it. And that will get resistance. With this approach, it's much more sustainable. And in answer to the question about resistance, I explained how we can deal with that in a rational way. So I think we have to explore these avenues. And I hope that young people like yourself and your students will think about these ideas and try and work out new ways for using the science which we have, particularly the genome editing, which I use in my own lab, which is a very valuable tool. But I think we have to show that there are real benefits for the public, whereas they perhaps think that BT cotton and Roundup Ready soya is not very interesting for them, only for farmers and for big companies. So we have to persuade them that maybe there's something in GM and the new technologies for them. And I think it's a job we must all embrace.
Of course. Thank you very much.
Thank you.
Professor Pickett, thank you very, very much. I'd like to let you know that we have more than 170 views live. This number will grow up by many, many times as the days go by, because the students are either taking class at the moment remotely or taking some other activities. But it was a wonderful talk, marvelous talk. Thank you very much for talking and explaining so clearly, in a very nice space, so that our students actually and everybody could really understand and appreciate the science that you presented to us this afternoon. Thank you very, very much. And I will just like to invite everybody within maybe 25 minutes to the section for the biological area. We have now a section. There's some selected works. They've been pre-selected because out of hundreds of posters that were presented. And there's an external panel from faculty from different schools. They will interview them and have a chat with them in 3 separate sections now at 4:30.
Well, in saying goodbye then, I must say I've really enjoyed this opportunity. I love coming to Brazil. I hope soon I will be able to return. I may start to meet some of you. We can talk more about this science, but I would like to offer congratulations to the prospective winners from this event that you're going to go to now. Goodbye for now. Goodbye and thank you.
Bye. Thank you. Bye.
Thank you.